This video is about GPCR, specifically about the role of phospholipase C. And I've called this video the 2-for-1 because, as you'll see, there are two different signaling pathways that are queued up through the activity of this one enzyme. Also, I wanted to break down this learning outcome a bit more, so before class, what I'd like you guys to be able to do is to be able to label the components of figure 16-27. Basically, I'll provide you with a figure that only has shapes and arrows, and then you'll have to label what the different components are, but there'll be no text on it. We're going to start off by looking at just a small portion of figure 16-27. It's really jam-packed with a lot of info. So let's start off with this part here, which is to check out phospholipase C. And so make that name work for you. You'll notice that this is working with some molecules which are embedded in the cell membrane, so that might help you remember the lipase part, and then you'll also see that some of these molecules have phosphate groups. And basically phospholipase C is activated by GPCR signaling, and then it will use inositol phospholipid and break that down into two different parts. And so this is a pretty complicated structure of this inositol phospholipid. So let's take a minute to break that down. Basically, there are two fatty acid tails, and then there's a glycerol. And that sounds a whole lot like a pretty typical phospholipid. But then we take it up a notch, because it is bound to this inositol triphosphate, and so that's kind of a complicated structure too. Basically, this inositol 1,4,5 triphosphate is named because we have a phosphate group bound to the 1, 4, and 5 position of this 6-carbon sugar. And as you may recall from other classes, we count carbons and we can number them. So this is the first carbon, then we have second, third, fourth, and then the 5th carbon. Hence the 1,4,5 triphosphate, and there's 3 phosphates. And again, this is abbreviated as IP3. So that's for the inositol phosphate, and then the 3 for the 3 phosphates. So kind of to sum up, this whole thing is the inositol phospholipid. This part here is the IP3, and this phospholipase C enzyme is going to cleave right here in this position to cut the IP3 off of the glycerol with the 2 fatty acid tails. And so that's what we see happening right here. So this is what's being catalyzed by the enzyme. And we call this phospholipid diacylglycerol, or DAG. And then this is the IP3 over here. You don't need to know the full names of IP3 and DAG. I think it's useful if you know them. You should know this name, inositol phospholipid. There's not an abbreviation that we're going to use for that. And again, why this is so interesting for us is that each of these products, DAG and IP3, are second messengers, and they're each going to go off and activate something different in this signaling pathway. Okay, so now that we have seen this small portion of the pathway, which is right here, we're going to talk a little bit about what each of those second messengers will trigger. Also in this figure, you can see the previous events that took place, and so let's not overlook those. Basically, we've got a signal molecule that binds our GPCR, activates it. Then, as you know, the heterotrimer is activated, and in this case, the gamma beta subunit is what will activate the phospholipase C. And they call this activated G protein, this gamma and the beta GQ. And that doesn't actually really stand for anything that anyone knows of anymore. You could think of it as Q for quirky. I guess the X and Z and some other common letters were taken, and so it just went for Q. There you have it. But basically, again, this is called GQ, the gamma beta subunits, as they activate phospholipase C. Note that phospholipase C is hanging out really close to the membrane. It's actually a membrane-associated protein, and so it's kind of moving around here. This membrane ends up being a pretty awesome meeting place for a lot of the G protein coupled receptors and other and the G protein itself. So after phospholipase C's activity, then there's kind of a fork in the road, and one of the forks that will follow is the IP3. And what's kind of curious here is that IP3 is going to act as a ligand and bind to the calcium channels, which are in the endoplasmic reticulum. A little backstory is that calcium is found at very high levels in the ER, and in particular in muscle cells. The ER is called the sarcoplasmic reticulum, and it has incredibly high levels of calcium ions. When IP3 binds to this channel protein, then it opens it, and that means that the calcium, which is at high levels inside the ER, will flow out down its concentration gradient. So now there is high levels of calcium, which are floating around in the cytoplasm, and this is going to diffuse over to this PKC. Notice this is not PKA, it's PKC. So PKC is protein kinase C. And this needs two things to be activated. The first thing it needs to be bound to calcium. And the second thing it needs to be bound to DAG. And so basically what happens here is that we've got this little kind of extra step for IP3, and DAG is still hanging out as one of the products of this enzymatic reaction. And so DAG is hanging out, it can bind to PKC, then we've got this little side road for IP3, the calcium comes out, and then when these two things bind together, then PKC is activated. And that can go stir up a whole bunch of other downstream targets and cellular responses. Okay, so I'm going to summarize what all went down with that, and this is what you guys should know for the exam eventually. Alright, so again we've got our plasma membrane, 7-pass transmembrane receptor. When it's bound to the signal molecule, G protein is activated. This time we're going to follow the gamma beta. And basically they will activate PLC, phospholipase C. Then PLC breaks down the inositol phospholipid into DAG. And IP3 makes a little detour over to the ER. This increases calcium concentration in the cytoplasm. And then that calcium can go diffuse over here where DAG has bound to PKC. Be super careful about all these PLC, PKC, PKA. They all sound a lot alike, but they each have different roles. And so we've got the calcium hanging out over here. Altogether, that forms the active PKC, which can go trigger some other targets, downstream targets, and elicit some cellular responses. So I thought I would tell you a few different ways that I try to remember the pKa, PLC, PKC. Totally optional, but this is my little mnemonic device. So pKa, that's a phosphokinase A, and so I like to think of it as kind of a type A personality. And it's basically lurking around in the cytoplasm of a variety of different cell types. As we saw before, in skeletal muscle, it can set up a kinase cascade, including phosphorylase kinase and glycogen phosphorylase. On the other hand, in neurons, it can head into the nucleus and phosphorylate the transcription factors. So to me, that kind of seems like real outgoing, and then depending on the particular environment, in this case the cell, it can go off and do a variety of different things and set up a bunch of other events to occur. So that's what I think of for pKa. PLC, I really try to remember that lipase part of it, so the phospholipase. And it helps me to remember that it is acting at the membrane, and in particular it is acting upon the inositol phospholipid and cleaving that into DAG and IP3. So that I think of that particular step. Then for PKC, I think of it as kind of like a complicated cat. I don't know, it's just what I came up with. Namely that this protein kinase C has two different components that are needed for its activation. And so that is over here, that as you know it needs the DAG and the calcium. You could also remember the C from calcium has to do with the PKC. But somehow don't forget that you're going to need to separate the PLC and PKC. And then just kind of take home point there is that be real careful with these three different components. They all show up within this GPCR signaling, and they have very similar names, but they have different functions. And so in this case, I think it's actually really useful to learn the full names of these enzymes because it's easy to accidentally put a different letter in there for the acronyms. And so that would just be a double check as you were answering questions and working through problems.